Now, let us continue our discussion regarding cell physiology. Next organelle we will discuss, that is lysosome. Lysosome is either round or oval membrane bound vesicle. Okay? And they are produced from Golgi apparatus and dispersed throughout the cytoplasm. They are present throughout the cytoplasm. They are like membrane bound vesicles. Okay? Size of the lysosome is about 250 to 750 nanometer diameter. Okay? And one of the important characteristic here is lysosome has thickest covering membrane, and that is again made up of lipid bilayer. Okay, lipid bilayer. Why it is important? Because lysosomal enzymes, they may destroy the other organelle. Now this lysosome, you can see they have many granules, and these granules, their size is about 5 to 8 nanometer diameter, and these granules, they contain various enzymes. These enzymes are hydrolytic enzymes. Okay, and these enzymes are also known as acid hydrolase. Okay, they destroy by in the acidic medium and they cause hydrolysis. Okay, they act best at the acidic pH. Okay, and pH inside is kept acidic by the action of the H plus pump or H plus ATPase. Okay, and these lysosomes are present in large amount in the cells having phagocytic activity. Which are our phagocytic cells? Important one, they are, one is neutrophils and other are macrophages. So they have large amount of macrophages. They have large amount of lysosome. Okay. Now, what are the functions of lysosome? Number one, they cause degradation of macromolecules, large molecules. First, they are engulfed by cell. Cell causes phagocytosis of that molecule, either by endocytosis, that is one type of endocytosis. And now what happens, suppose this is the macromolecule which enters inside the cell. Now lysosome fuses with this macromolecule. Okay, and it releases this was primary lysosome first, now it is converted into secondary lysosome, and pH of the secondary lysosome is now becoming acidic, where lysosomal enzymes, they become active and they are released inside the macromolecule and it is destroyed. So that is first function of lysosome. Second function is worn out organelle. Rough endoplasmic reticulum also we have discussed. They provide wrapping around the worn out organelle, but again this wrapped worn out organelle is going to the lysosome where it is destroyed. Okay, so that is another important function of lysosome. If there is excessive amount of secretory product produced by the cell, that is also destroyed by lysosome. And last but very important function is autolysis. After cell dies, these lysosomal enzymes, they cause autolysis. Whatever remnant particles are there, they are destroyed by lysosome. Therefore, these are known as suicidal bags. Okay. So these are the four functions. Let us repeat: degradation of macromolecules, degradation of worn out organelle, excessive secretory products are removed, and autolysis. Okay. Next organelle, that is peroxisome. These are also similar to lysosome, but lysosomes are produced from Golgi apparatus. These are produced from smooth endoplasmic reticulum directly. Okay, and these are prominent. They are present in large amount in liver cells. Liver cells are known as hepatocytes and tubular epithelial cells. They have large amount of this peroxisome. Okay. Now, another important difference between lysosome and peroxisome is lysosomes, they contain hydrolytic enzyme, whereas here they contain oxidative enzymes. Okay, which are these oxidative enzymes? Either they contain catalase, urate, oxidase. Okay, so these are the oxidative enzymes, and oxidation causes breakdown and removal of hydrogen peroxide as well as free radicals. They also contain catalases, which acts on the hydrogen peroxide, and by acting on the hydrogen peroxide they liberate oxygen. Now, what are the functions of peroxisome? Number one is they degrade toxic substances. Which are the toxic substances degraded? Mainly toxic substances like hydrogen peroxide or metabolic waste products. That is one function. Second is they are the major site for the oxygen utilization because oxidative enzymes are there, so they require oxygen. Okay. Third, that is if excessive amount of fatty acids are there, they are broken down by process known as beta oxidation. And fourth, that is gluconeogenesis, means formation of glucose from non-glucose sources, that is gluconeogenesis. So here it is produced from fat to glucose. This is the process which is enhanced by the peroxisome. Okay. Another important thing here is alcohol is detoxified by the peroxisome, mainly in the liver cells. So large amount of peroxisomes are present in the liver cells. Next organelle, that is centrosome. Structure, these centrosomes, they are two short cylinders, like cylinder-like structure, this kind of two short cylinder-like structure. Okay. Now, and they are visible only during cell division. They are located mainly near the nucleus, because at the time of cell division, when there is a movement of chromosomes, that follows the movement of centrosomes. Okay, and they are located at the right angle to each other. And this cylinder shaped your centrosome, they have microtubules here in their wall. Okay, and these microtubules are in the group of three, like this 1, 2, 3. So they are the triplets, and this 9 triplets are there. Suppose if this is here 1, so this triplet 1, 2, like this, they have 9 triplets in, this is the cut section you would say. Okay. So this triplets, they run longitudinally and 9 triplets are present. Okay. And they have microtubules. Okay. And if centriole is absent, cell cannot divide, and this is the reason why neuron cannot be divided, because neurons don't have centrosomes or centriole. Okay. So during cell division, these centrosomes, they have important function. Next organelle, that is secretory vesicles. These are the organelle again having limiting membrane like lysosome. They have secretory substances, and these secretory vesicles are produced from the endoplasmic reticulum first, and then their processing and packaging takes place in the Golgi apparatus. Okay. So they have again vesicle, they have secretory substances, and whenever these substances are required, this vesicle ruptures and the substance is released. Next organelle, that is mitochondria. Mitochondria, other name is powerhouse of the cell. Why? Because it produces energy for the cell. Now, what is the structure? It is either filament, like filament, or like a globule, filamentous or globular in shape, and its length is about 5 to 12 micrometer, diameter is about 0.5 to 1 micrometer, and here it is a major site for aerobic respiration. Aerobic respiration that produces large amount of ATPs, and this is the reason why mitochondria is known as powerhouse of the cell. Now, this is the structure of mitochondria. It has bilayered membrane. So it has 2 layers of the membrane. Outer membrane, this is outer one, this one. It is smooth, okay, and it has enzymes for biological oxidation. And inner membrane, you can see here, it is folded, and these folds, they are known as cristae. If it is one, it is crista, plural is cristae, and they have enzyme ATPase as well as other enzymes which are required for ATP synthesis. Okay? And inside mitochondria has got matrix, mitochondrial matrix, that is fluid, and it contains enzyme for TCA cycle, citric acid cycle, as well as respiratory chain oxidation. Now, what are the functions of mitochondria? Number 1, as we have discussed, it is powerhouse of the cell, so it produces energy. Okay? Mitochondrial enzymes, they cause oxidation of the nutrients, and energy is released, as well as carbon dioxide and water, like in Krebs cycles we have discussed. Okay, so ATPs are produced. Second important function of mitochondria is mitochondria, they are self-replicative, means similar to DNA of nucleus of the cell, mitochondria also contains its own DNA. Okay, and it plays an important role in the replication, means one mitochondria can produce another one, and replication takes place when ATP synthesis requirement of the ATP is increased. And third thing, this mitochondria is derived from the mother, so it is the DNA for the mitochondria, that is maternal. Next is nucleus. Nucleus is present in the cells. Those cells, they divide as well as they produce enzymes. So nucleus has mainly two functions. It is required for division of the cell and for the production of protein, mainly enzymes. Okay? So nucleus is controlling center. And according to presence and absence of nucleus, cells are of two types: prokaryotes, they don't have nucleus, and eukaryotes, they have nucleus. Most of our cells, they have single nucleus except skeletal muscle cells. Skeletal muscle cells, they have multiple nuclei. Yes, multiple nuclei. Okay. Now, what is the structure of nucleus? Nucleus has got nuclear membrane outside, then inside there is nucleoplasm. It is different from the cytoplasm, and it contains nucleolus. We'll discuss. Starting with nuclear membrane. So this is covering nucleoplasm. It is double layered membrane and it has got pores. You can see here it is like this. We can draw it like this. It has got pores like this and it is bilayered. And this one here you can see like this. We can draw the membrane. Okay, this way. We'll discuss why these gaps are left here. So this is nuclear membrane. Okay, so bilayered porous. Okay, that is thing. And this pores of the nuclear membrane, these pores, they are about 1000 angstrom in diameter, and they are covered by thin homogeneous membrane, which allows passage of macromolecules like RNA pass from nucleus to the cytoplasm through these pores. Okay. In between these two layers, there is a space, this space. It is known as perinuclear cistern. Okay, perinuclear cistern. And through pores, this cytoplasm and nucleoplasm are in contact. Okay. Now, another important thing here is outer layer. This you can see it has got two layers. So, this outer layer of the nuclear membrane, it is continuous with, we have what is here? We have endoplasmic reticulum. So here it is continuous with the endoplasmic reticulum like this. Okay. The space between these two layers of the nuclear membrane is, this one is 300 angstrom, this space, and the nuclear pore, that is 1000 angstrom. And this is, as we have discussed, perinuclear cistern. And it is continuous with the endoplasmic reticulum, this one. The space is continuous here. Then next thing is nucleoplasm. It is not fluid like cytoplasm. It is gel-like substance. And it has genetic material in the form of DNA. And DNA forms genes. During cell division, this DNA and this chromatin, which is converted into chromosomes. So chromosomes, they become visible. And in between cell division, we have chromatin. Now, what is this chromatin? That is like thread-like this material, which consists of large molecules of DNA. This is chromatin, not chromatid. Chromatid, that is arm of the chromosome. So don't get confused here. Okay. And third thing, chromosomes we have discussed. Third thing, that is nucleolus here. Inside the nucleus, one or more nucleoli. If it is one, that is nucleolus. If it is more, it is known as nucleoli. Suppose two are there. So one or more nucleoli are present in the nucleus. Okay. And it is prominent and very numerous in large amount if the cell is growing. And from this nucleolus consists of two things: RNA plus protein. RNA is synthesized and stored in the nucleolus, and it is transported, you can see, through the pores. It gets transported to the cytoplasm. Okay. What are the functions of nucleus? Nucleus is there in the center, so it controls all the activity of the cell. Okay. Which activities you can include? Like commonly metabolism, growth, and protein synthesis. Metabolism, growth, and protein synthesis. So for control of this activity. Second is reproduction and multiplication. For that, definitely nucleus is required. If there is no nucleus, no reproduction. RNA synthesis. RNA synthesis. For RNA synthesis, we have a DNA. DNA acts as a template from which messenger RNA is produced, and through pores this messenger RNA comes out, and it is important for protein synthesis. So that is important for RNA synthesis and protein synthesis also. Then genetic information is transferred, storage and transformation of hereditary information. That is also one of the important function of nucleus, and it also controls cell division. At the time of cell division, these chromosomes, they are going to different poles, and that is important for cell division. So these are the important functions of nucleus. Ribosomes are either free or they are attached on the endoplasmic reticulum. So they are dot-like structure, and their diameter is about 15 nanometer. They are either attached with the endoplasmic reticulum or they are free, and main function is they are concerned with the protein synthesis, whether the protein is a hormone or enzyme or mitochondria. Any protein synthesis any protein synthesis requires ribosomes. Next we'll discuss about cytoskeleton. Cytoskeleton, that is skeleton of a cell. Like in our body we have skeleton. So cell has its own skeleton, that is cytoskeleton. It consists of complex network of fibers. Okay? And mainly three types of cytoskeleton that is there. One, that is microtubule, second is intermediate filament, and third is microfilament. Major function is they provide shape of the cell, they give support to the cells, and they are also important for movement. Like if we don't have bones, we don't have shape. Okay? In our body, same way cells, they also have shape because of cytoskeleton. Starting with microtubules. Microtubules, they are straight as well as hollow tube-like structure, and they are made up of the protein. Name of the protein is tubulin. This tubulin forms the microtubule. Okay? And the diameter, so microtubules, they are the thickest one, so their diameter is 20 to 30 nanometer, and they form cilia of the cell, flagella of the cell, they form centrioles, and they are important for various functions, and they have this tubulin. This tubulin are of two types, alpha and beta tubulin dimers, and the length of the microtubule is changed by either addition or removal of the tubulin. So it is like this tubulin dimer, like this. If this dimer is added, the length increases, and if it is removed, then length decreases. So microtubule organizing center exists near the cell's nucleus, and this microtubule, they grow out from the center towards the periphery, and this microtubules, they move. What is the function of the microtubule? They are important for the movement of intracellular vesicles. Like if vesicle has to move from one part of the cell, here this is the cell, to other part, so they move by sliding on the microtubules. Okay? Now this movement, it is driven by motor proteins. These motor proteins are of two types. One is kinesin and other is dynein. Now what is this kinesin and dynein? So is this, you can see this microtubules, they act as a track. If we have a railway track, so the goods will pass here, but for movement we require engine also. So these are driven by the motor proteins, kinesin and dynein. So kinesin, what does it do? It causes transport from the center of the cells towards periphery, this side. Suppose we have a train which moves the goods. So this train is moving away from the center, kinesin, and dynein moves towards the center. So this microtubule acts as a track, and kinesin and dynein, they are important for the movement of the particles. So they are known as a molecular motor. Okay. Now what are the functions of microtubules? So first, that is they determine shape of the cell, provide structural strength. They are also important for the transport of granules, transport of substances. They also make cilia and flagella, spindle. At the time of division of cell, you can see here. At the time of division of cell, you can see here, these microtubules, they start from this centriole like this, and chromosomes, they move to the periphery. So this is also one of the important function of the microtubule. Next are intermediate filament. The diameter is less than microtubule. Microtubule diameter is 20 to 30 nanometer. Here this is 10 nanometer. Okay, and here difference between filament and tubule. Tubule is hollow like this. There is a gap inside, and filament is like thread. Okay, so that is filament structure. And what are the functions? Mainly it connects the nuclear membrane with the cell membrane. Suppose this is a cell. This is the nucleus. So nuclear membrane with the cell membrane connection is by the intermediate filament. So what are the functions? They integrate the cell organelle within the cytoplasm, also maintain shape of the cell, and they connect interior of the cell to the adjacent neighboring cell, yes, through desmosome and hemidesmosome. So they provide strength to this desmosome, hemidesmosome we will discuss. And if they are absent, the cell will rupture easily. Yes, so this is important function of the intermediate filament. Then comes microfilament. They are, you can say, diameter is least. No? Microtubule 20 to 30, intermediate filament 10 to 12, and this has the diameter of about 3 to 6 nanometers. So they are again filament, like filamentous structure, thread-like structure, and they are made up of non-tubular protein. Microtubules, they have tubular protein, tubulin. Here these are made up of non-tubular contractile protein, actin and myosin. We all are familiar with the term actin and myosin, and they have this actin myosin contractile protein, which is important for the cell movement, contraction of the cell. Also they provide structural strength, as well as important other function is they are important for the phagocytic action by WBC. One applied physiology here is this microtubules. They are target of number of anti-cancer drugs. Some anti-cancer drugs, if they destroy this microtubules, then the cell division of this cancer cell is not possible. So this are targeted by the drug. For example, like one drug is vincristine. Vincristine, it decreases polymerization of tubulin, polymerization of tubulin dimer. As we have discussed, tubulin dimers, assembly and removal of this dimer, that changes the length. So this, if polymerization is reduced, means there is no formation or decreased formation of microtubule, and if microtubules are not there, there is no spindle formation and no chromosomal division and no cell division is possible. So this is the one action of the drug vincristine, that is anti-cancer drug. Other is Taxol. It is also an anti-cancer drug, but its action is it stabilizes microtubule. Stabilization of microtubule also again decreases mitosis. Another applied, that is Carter-Jenner syndrome. It is an autosomal recessive disorder. Here dynein is missing in the cilia. So if dynein is absent, then dynein is a molecular motor, so movement is absent. So if ciliary movement is absent, that results in recurrent respiratory tract infection, because cilia of the epithelial cells of the respiratory tract is important for the removal of foreign particles. As well as in males, sperms, they have flagella, and their movement also requires dynein. So if dynein is absent, flagellar movement is also absent in the sperm, and that results in infertility. So the symptoms are the patient is having recurrent respiratory tract infection because of lack of movement of cilia, and because of lack of movement of flagella, male patient is having infertility. Cytoplasmic inclusions. These are not organelle, but in some cells these inclusions are present. Sometimes they are present or maybe absent also. Now, which are these inclusions? One, that is glycogen. It is present in the liver cells and skeletal muscles. Fat is present in the adipocytes, liver cells and adrenal cortex. Proteins, they are present in the secretory granules, in the secretory glandular cells. Melanin pigments, they are present in the epidermis as well as in the retina and basal ganglia. And lipofuscin, that is yellow-brown pigment, which is derived from the secondary lysosome, and that is present in the brain cells as well as heart cells, and commonly it is present in elderly individual. If the individual is well fed, overnourished, liver cells, they also contain glycogen or adipocytes, and during starvation they disappear. So this is all about cell physiology. Thank you.
If you wish to get all topic notes
My book Physiology Notes is available on Flipkart as well as Amazon, and link for both that is given in the description box. And to get the soft copy, my application Vesalius that is available on Play Store, you can download it and get the subscription. Link for that is also given in the description box. Thank you so much.